Hello everybody, I hope you guys had a great long weekend. Welcome to Concept 2 Notes. This is going to be about homeostasis and regulation. Should start, I think it's on page 19 in your guys' packets. So if you want to flip there, make sure you have those open so you can follow along. Again, this is all, or it should be at least review from biology, but this is going to be some foundational stuff I want you to have down. So just make sure you're following along with the packet. All right, so first off, here's your definition for homeostasis. Basically, it is a stability of the internal environment and the mechanisms that maintain that stability. There are different types of equilibriums that you're going to see. So dynamic equilibrium is maintained where the rate of loss balances out with the rate of gain. And we call this a dynamic equilibrium because it is always changing. So there's typically a range that we would like different things to stay within for our body. So usually it's not going to be always one specific number, but there's kind of a range. There's a high end and a low end that we want it to stay between. So that's why it's dynamic and not static. Organisms are going to detect and respond to different stimuli that our bodies encounter, and that is going to be what creates this dynamic equilibrium. And homeostasis is maintained through regulation at the organ system level all the way down to the cellular level. So at the bottom here, there are three different examples that you can see. So first one is going to be pH, and you can see there is a acidic side and a basic side, and we want to be kind of in the middle here. So that's the range for pH. For temperature, obviously it's going to be either cold or hot. And then for blood sugar, it can either be hypoglycemic or hyperglycemic. So here you can see these are just three general examples. There's plenty more we could come up with. These are just three general examples of how of different factors that must be kept within a specific range in order to help maintain homeostasis. All right, so feedback mechanisms, these have evolved to help maintain homeostasis in organisms, and there are basically different loops that are going to use the output of a system to signal a change in input so that a system response can be stabilized or amplified. Basically, it's just going to be how you see here. There will be a stimulus, that's the input. It's going to affect the system, which is going to be usually a specific organ, and then that organ will then communicate with the rest of our body, causing the output, which then kind of goes back and back. So that is why it is called a loop, and these are going to be different feedback mechanisms that our body uses. These mechanisms can be either positive or negative. So for positive feedback mechanisms, the output or the product of a system intensifies the response. So you can see here, this is going to be amplified. So as our body gets the stimulus, it's going to send a message to that specific organ, which is then going to send a message throughout the body to create the desired response. And that response is going to be an amplified response. So a couple examples of that. First one could be with human childbirth. So as a childbirth process begins, the body is going to experience an influx of hormones that are then going to cause contractions, which is going to cause a lot of pressure in the body, which then releases more hormones. So you can kind of see how it's going through this loop here. So it releases more hormones and more contractions, more pressure, etc. And it kind of keeps going through that loop until eventually the child is born. Another example, not necessarily with a human, but with fruit. So when a fruit is ripening, it's going to release ethylene, and that ethylene is going to signal to surrounding fruit that it is ripened, and that will cause the neighboring fruit to ripen as well, which releases more ethylene. And again, it just kind of goes through this feedback loop, and it's amplifying the response. So that's a positive feedback mechanism. It's going to amplify the response. Then with a negative feedback mechanism or a negative feedback loop, the output or product of a system causes a counter response to return to a set point. So this is going to be our body receives a response. It causes the organ to create the output of whatever is needed to counter that response, and basically goes through this loop. So instead of amplifying, it's going to be a stabilization effect. So some examples with that would be like thermoregulation with human body temperature. So if our temperature gets too high or too low, there are different ways our body can regulate that, which we're going to talk about here in just a minute. Also with water concentration, that's osmoregulation. That's just another way that the negative feedback loops occurs, as well as with blood sugar regulation. So those are just 3 examples right there. Basically, you need to know with a negative feedback loop, this is going to occur when we are out of the desired range and we need to get back in the range. So think again with the human body temperature, when you get hot, you're going to start sweating, and that's going to be your output to kind of help your thermoregulation. So we'll talk more in depth with that in a second. So feedback loops, these are really an important way for us to see how the body can affect, or how our body can respond to the different receptors, stimulus, and effectors. So the first response is you're going to get a receptor, and this is basically a specific organ that in your body that's going to receive the stimulus. Once that organ receives the stimulus, it's going to evoke the actual stimulus, which is an action that will evoke a response within your body. That can usually be anything from sweating to releasing hormones, increasing your body temperature, all kinds of different responses that can occur due to this stimulus. And then the effector, this is the organ that does the response. So there's a receptor organ and then an effector organ. Usually they are different organs. And then finally, there's going to be a response, which is the effect that is caused by the stimulus. So you can see here, you're going to start with your receptor organ, and then you have 2 different scenarios. You can have when your feedback loop is too high. So if the system gets too high from its target goal, or if it gets too low from its target. So once it gets too high, you'll see it will go through this way, and then if it gets too low, kind of goes around the other way. So same kind of concept or idea, and I'm going to go through a few examples of different feedback loops that our body uses. So a really great example of a feedback loop that we're all probably really familiar with is thermoregulation. This is a really oversimplified view of thermoregulation, just FYI. But basically, you can think about so our normal body temperature in a human is going to be 37 degrees Celsius or 98.6 degrees Fahrenheit. And so when the temperature in our body is too high, it's going to be this top portion right here. Basically, the hypothalamus in our brain is going to be our receptor organ, which will receive the signal that your body temperature is above its normal range. And this is kind of this stimulus. So when our body gets the hot stimulus, our body is hot, that is going to initiate an action in the effector organ, which could either be your sweat glands or your capillaries. And that is going to cause a response, and your sweat glands will start to release sweat, which evaporates off your skin and will ultimately cool you down. So your capillaries are going to dilate to increase the overall surface area for heat to be released. And the overall response through this is that your temperature is hopefully now stabilized. So that's the example if it's too high. If it's too low, the hypothalamus, again, is the receptor organ, but it's going to receive the signal that your body temperature is below its normal range, so you're cold. And this cold is a stimulus that is going to initiate an action in the effector organs, which could be your muscles or your capillaries. So instead of sweat glands and capillaries when we're too hot, if we're too cold, it's going to be muscles and capillaries. So think about when you're cold, what your body normally does, and that's going to be, you will usually shiver. So the response is going to be your muscles will shiver, which requires energy. So ultimately, heat is released as a byproduct of this response, and also your capillaries are going to constrict to decrease surface area for heat to be contained and not released as much. So kind of the opposite effect that we saw when it was too high. And ultimately, again, your temperature should now be stabilized. So that was the example if it's too low. Another example with a feedback loop would be osmoregulation, and the normal osmolality of human blood is somewhere in this range. So just keep that in mind. Again, the receptor organ could potentially be the hypothalamus in your brain, or it could be the pituitary gland as well, as we'll see in a second. But so when your osmolality is too high, the hypothalamus in your brain is going to be the receptor organ that receives the signal that your blood is too concentrated, which means it's above its normal range. And this also means that the water level is too low and you are very dehydrated. So that is going to be the stimulus. And that is going to signal to the pituitary gland to release ADH, which is the stimulus that initiates an action in the effector, which is the kidney nephron. So that's the cells of the kidney. This causes a response in your kidney, and your kidney will retain more water instead of releasing it into your urine. Ultimately, now your osmolality is now stabilized. If it's too low, again, your hypothalamus is going to receive the signal that your blood is too dilute, meaning it's again below the normal range, and your water level is too high compared to the solutes in your body. And that is just going to signal to the pituitary gland to stop releasing ADH, which is the stimulus that initiates an action in the effector. And the effector is going to be, again, the kidney nephron. So this is going to cause a response in your kidney, and your kidney will release more water into your urine. And again, same result, you're now going to be in a stabilized range. Last example I have is going to be blood sugar regulation, which is one that is really important, and a lot of people, you're going to see this a lot in this class. So when your blood sugar is too high, which this is the normal blood sugar range for humans. So when it's too high, your pancreas, which is the receptor, is going to receive the signal that your blood sugar is above its normal range. Too high of blood glucose is going to be the stimulus, and that will initiate an action in the effector, which are the beta cells in your pancreas. That's going to cause a response. Your beta cells will release the hormone called insulin, and your body cells will then take in glucose from your blood, lowering your blood sugar concentration. Your liver cells specifically are going to take in glucose and store it as glycogen, and your blood sugar will ultimately be stabilized after that. On the other hand, when it's too low, and your pancreas, again, is going to be the receptor that receives a signal that your blood sugar is below its normal range. Too low of blood sugar, blood glucose, is now the stimulus that initiates an action in the effector, which is going to be the alpha cells in your pancreas. This causes a response of your alpha cells are going to release a hormone called glucagon. Your liver cells will break down glycogen and release glucose into your blood, raising your blood sugar concentration, and ultimately your blood sugar should now be stabilized. So another great example there. Feedback loops, when a mistake happens in a feedback loop, like just think about how we talked about with your blood sugar, if it's too high or too low, when that is thrown off, homeostasis is going to be thrown off, and your body is going to have to learn how to deal with that. So type 1 diabetes is a really common example that we're going to see of when a feedback loop does get a mistake in it. And I'm going to show you a video here in a second that is going to kind of review homeostasis, feedback loops, and some examples of when a mistake happens in a negative feedback loop, how that can result in diabetes. So just a couple more slides and then we'll get to that video. But Basically, enzymes have a very important role in homeostatic regulation. So, our living systems depend on reactions that occur spontaneously but at very slow rates. So, we already know that enzymes can act as catalysts, which are substances that speed up the reactions without being permanently altered. So, no catalyst will make a reaction occur that cannot otherwise occur, and most biological catalysts are proteins, which are enzymes. Every step in a metabolic pathway is catalyzed by a specific enzyme. So we kind of talked about that earlier, where like the enzyme is the lock, and different steps in the metabolic pathway are going to be the key. So each step is going to need that specific key in order for it to work. And remember also that enzymes are not used up in the reaction. They are still, they're unaltered. And again, they're not necessarily making the reaction go faster, but they are just lowering the activation energy required for the reaction to occur. So already talked about a couple of those things as well. Cells can regulate metabolism by controlling the amount of an enzyme. They can also turn synthesis of enzymes off or on. This is called feedback inhibition. The output of a process is used as an input to control the behavior of the overall process itself, usually leading to inhibition of the process. This is basically feedback inhibition helps with the cell's efficiency. It just is helping it not to waste resources. And so it's going to regulate different things such as the activity of an enzyme, and chemical inhibitors can also bind to enzymes in slow reaction rates, while environmental conditions can also play a role in enzyme function. So pH and temperature changes can lead to denaturation, basically meaning that enzymes will no longer be able to function properly. So remember, those enzymes have to be in their specific form in order for them to fit into the lock, or in the example we're using, into the, if enzymes are the key, they have to be able to fit into that specific lock. So if they undergo different changes, then they're no longer going to be able to fulfill their role. So that is all we are going to get through today. I am going to show you just a real quick video. Let's see if I can get there. Okay, I might have to pause this and come back. Hold on. Sorry.